All right, let's talk about our next system, the nervous system. So the nervous system is very complicated. We're still just barely understanding how it works and what happens as a result of different injuries or genetic disorders or diseases, um, or even just what's going on in certain areas of the brain and how different people can have parts of the brain be completely missing and yet still be completely functional. It's very complicated. And a lot of times in medicine, what you're actually doing is not dealing with the nervous system at all, but dealing with everything else, because once the nervous system becomes damaged, it's really hard to fix it, at least at this point. In a lot of brain surgeries, what they do is they have people, if they're, have an incredible talent, like violin prodigies, they'll have the person be awake during brain surgery, playing the violin or doing whatever it is they do that they can't function without. And the doctor will be working in the brain while this person is awake and playing the violin with just general anesthetic to make sure whatever part of the brain they're going into won't affect that part of the brain that is is doing the work that person has that's so unique. Which seems like such a barbaric, primitive thing. Like, how about I work on your brain while you're awake? It's cool, just play your violin. But it's the best we have right now, and it's a lot better than what people used to do. Stuff like lobotomies, where they would shove ice picks in people's noses or in the orbital cavity, and then just kind of shake that ice pick around in the front of the brain to get rid of any unwanted behaviors, such as autism or sexual deviancy, or even just speaking out. But what it did is actually just disconnected all the neurons in the part of the brain that give you personality. And so, you basically ended up with a zombie type person. So we're slightly ahead of that now. But it's still the nervous system is still incredibly complicated. But there's some basic stuff that we do know that everyone needs to know, especially for this course. And one set of those things are the functions of the nervous system. So we know that the nervous system takes in sensory input. So anything coming at you from the environment or anything gets received by the nervous system and either processed by the brain or by the spinal cord. So it receives sensory input, processes and integrates this information, and then sends out commands. It seems so simple, and all of this is in order to maintain homeostasis. So you hopefully remember this word. Homeostasis is just the state of your body keeping everything so that you stay functional. It's trying to keep everything the same. So if you get cold, homeostasis is going to be maintained by making you get warm. And that could be causing you to shiver, that could be causing a behavioral shift, so you go put on a sweater, whatever it is. So whatever the brain is doing, it's trying to get to make sure you maintain homeostasis. You maintain whatever internal state it is that keeps you functional. So I want to mention the references I use for this lecture. There's some really good ones. So I use a lot of the figures from the OpenStax Anatomy and Physiology book. So this book, if you haven't been to the OpenStax website, they are amazing. I don't have a link open right now, but this is put out by Rice University in Texas, and what they do is they have free textbooks for so many different subjects. So they have them in maths and sciences, social science, humanities, even AP courses. And so I'm using the anatomy one for a lot of the lectures here. They're just PDF downloads, and they're really, really good. So I highly recommend it if you need a textbook. They do go above and beyond what we're doing in this course, but they're still good for just a general review. Okay, let me move this back. There's this he was a medical student, and I think he's beyond that now, but he makes these amazing videos. I used this guy, what is it? Oh, here he is. Armando Hazadungan has a really good set of medical videos. If you want to go watch them, it's the whiteboards type where he draws it out while he's talking, and he's an amazing artist. And then I use this neuron animation too. So just so you have all of these links. So let's talk a little bit. So we're going to split the nervous system into two very overgeneralized lectures for this class. And in the first one, we're talking about what makes stuff happen in the nervous system. So if this is the brain, this is a spinal cord, and you're in your friend's shower trying to figure out how to make it work because it's really confusing, and you're trying to figure out, I turned on the water, am I going to need to heat it up or cool it down? Now, if the water turns on and you're taking in that sensory input, which here is in blue, these are not blood vessels, these are neurons, okay, or these are nerve tracts or nerves that are taking in a signal. Say, oh, sensory changes, what temperature is this? If it's way too hot, like boiling hot, you have a reflex. It's going to make you jerk your hand and contract those muscles. And what happens is that sensory neuron comes in, it actually skips the brain completely and actually connects directly with the motor neuron, which causes you to jerk your hand, and that's a reflex arc. Now let's say it's slightly too cold, but not like reflex worthy, and so you're taking in all this information and it's going to your brain and saying, do I make something happen? Am I gonna, so it integrates that information to say, okay, this is too cold, I want to heat it up, what do I do? And then it sends a signal there. When it decides on something, whether you decide or it's your brain deciding, however, we won't get into the philosophy of it, it sends a signal back through the motor neuron pathway that says, why don't you grab that knob and make it hotter or colder, whatever you need to do. So we understand that there's these pathways, but what actually makes up those pathways? And that's the neuron. And so we're going to look very into the detailed anatomy of the neuron. Okay, so let's take a second to review some cell biology and then look at the separate parts of the neuron. Okay, so way back in Module 3, we talked about a basic cell. Okay, so we talked about how it has a cell membrane, that basically what we described it as is just what contains the cell and keeps it separate from its environment. It has a nucleus, which contains the DNA, which is the control center for the entire cell. And then there's a bunch of other stuff happening. We're just going to talk about these two things right now because they're relevant to the neuron, the cell of the nervous system. But we want to expand a little bit on the idea of the cell membrane. So the cell membrane not only keeps the cell distinct from its external environment, the extracellular matrix, but it also acts as a control, like a doorway basically, to make sure whatever's outside can't get in, whatever's in can't get out unless it's supposed to. So we talked a bit about diffusion, and so diffusion is whenever you have a whole bunch of stuff in one place, it wants to spread out as much as possible. Well, once it hits the cell membrane, it has to, it's only going to diffuse if it's allowed to. Like, first of all, if it's a small charged object, like an ion, it might be able to diffuse, depending on how small it is. So if I have a whole bunch of sodium here, Na, so sodium is abbreviated as Na, and I only have like one sodium inside. Well, there's way more sodiums outside, so they're going to be able to diffuse into the cell until it's even, okay? So, because they're going to move to where they're least concentrated. Now, if I'm doing that in the way that it's going down the concentration gradient, so from where it's more concentrated to where it's less concentrated, that takes no energy, okay? So if you're going with the concentration gradient, you're diffusing in the direction of from where it's more concentrated to least concentrated, that requires no energy. Now, so this is a review of Module 5, by the way. If I instead want this sodium to leave the cell, well, that's going against the concentration gradient, and that's saying I want something that's in a place that's not very concentrated to suddenly be moved to where it's really concentrated, and that will take energy. And this is going to become really relevant when we talk about the neuron, because that's how the entire thing works. So let's say we take this cell, and I have a fancy visual. We take this idea of a cell that has a nucleus, and we're going to take that take the cell membrane of it, and we're going to start stretching it out. Okay, so if I take that cell membrane like this, and I start stretching it out into these weird little portions, and they kind of look like trees, and the idea is that these big projections of the cell membrane can then talk to other cells, they can interface with them, and then I take one part of that cell membrane and I really stretch it out like it's silly putty, and I'm just stretching and stretching and stretching, and then the very end I stretch out again into more little tendrils. This is a basic multipolar neuron, or a basic neuron. Okay, so let's look at the different parts of the neuron. So it's the same idea that we have this general, here's the cell, okay, so this is literally called the cell body, but I've stretched out parts of the the cell membrane at the top, and the idea is then it can interface with lots of other neurons. Okay, we're going to talk about what that's called. So you can have one that's kind of interfacing here, you can have another that's kind of interfacing here, and so all these different tendrils can connect to different neurons in the system. And we see that people who are very active academically or intellectually or creatively have a lot of connections in these. These are called dendrites, these extensions of the membrane. And people who have dementia or Alzheimer's tend to, or depression even, they don't have a lot of these. They tend to, these go down a lot, and just if there's not a lot of neural stimulation, your body's going to get rid of what it considers extra, and that can lead to a lot of issues with intellectual capacity and memory, and lead to Alzheimer's and things like that. Okay, so that's the neuron. It's a different kind of cell. So let's kind of take this and label the different parts. And again, I took this from this amazing artist Armando Hazardungen, who does a lot of medical videos, and I highly suggest you check his stuff out. It's very, very good. So let's talk about the different parts of this neuron. So you'll notice this one has these wrappings. So each one of these is actually a cell. We're going to talk about what that is in just a second. But let's say, okay, this part of the neuron contains the cell body, or the soma. You may see it called a soma. And that's this part here that contains the nucleus. And then it has all these little extensions, and those extensions are called dendrites. Okay? And what the dendrites do, again, is they connect to other neurons so that you can pass signals down. Because one of the defining characteristics of a neuron is that it's directional. Okay, so neurons are directional, and they're polar. So I don't know if we'll consider, they're definitely directional. We'll just go with that. So the idea is a signal will always propagate or move from the cell body down this part, which is called the axon. Okay, so this is the axon, to the axon terminal. Okay, so this is the axon terminal. And again, we have more of these aren't dendrites, they're just extensions of the axon terminus, the axon terminal, which that can then end on something and release a signal. So when we were looking at the neurons, when we were looking at the muscles, these were ending on a muscle cell. So if I have a muscle cell here, this would then be the neuromuscular junction, and this neuron would be releasing bloop, bloop, these neurotransmitters. And in the case of the neuron, they would be ACTH. Okay, so how does a neuron know when to signal something like a muscle to contract? Well, and how does it actually do this? There's a lot of different things going on. So before we move on, let's talk about what are these things, okay? So the whole way that this thing works, the neuron works, the way it sends a signal from here all the way down the axon to wherever it's going to synapse, okay? So when neurons connect, it's called the synapse. So this space between the neuron and whatever it's connecting to is the synapse. Okay, it could be connecting to other neurons, like I showed before. I could have a neuron here instead. It could be connecting to a muscle. It could be connecting to anything. Okay, you also can have more than one neuron synapsing. So, okay, another thing to remember is that, or to know, is that the synapse is just wherever one neuron ends and something else begins. So if I have a neuron coming in and synapsing with this neuron, this is a postsynaptic neuron. This would be the presynaptic neuron. But if this is a long chain of neurons and there's also a neuron here, well, in that case, it's kind of hard to, I can't really zoom out, but in that case, I have a presynaptic neuron in this case, because that would be a synapse. And then once I get to the end here, this is now a presynaptic neuron because it's synapsing with this neuron. So you can have really long chains of neurons, but the signal can get lost a little bit that way. And also, if you think about it, if you have a bunch of neurons that look like this, that are all sitting here, and you're somehow propagating a signal down these axons, you don't want that signal to kind of leak out into the environment and interfere with other neurons. So that's what these yellow things are. These are Schwann cells, named for the guy, oh, there's a cat. All right, back up. They're named for the guy who discovered them, or at least first labeled them. And what these are very, very flat cells that wrap themselves around, so you can see here's the nucleus of this cell. They wrap themselves around this long extension of the membrane here, so that it basically acts as a insulation. So it's like copper wiring. If you have wires just like bare, like let's say your iPhone charger or your phone charger, that little part before the charging bit starts to get old and starts to break, and you can see the wires inside, and sometimes you see them spark a little bit. Well, that means your signal is being lost. It's not being transmitted completely. It's getting lost out of the side of it. Well, you don't want that to happen and have your other neurons around you be receiving the signals. So something like
oh my goodness, there's Parkinson's disease. In Parkinson's disease, these Schwann cells start breaking down or they don't get fixed. And that signal from one neuron that's going and saying, hey, don't contract, stop contracting to the muscle or whatever, suddenly that signal leaks and that signal goes to a neuron over here that's saying, no, you should contract, and you can't control your muscles. And so that this insulation is really important to make sure the signal that's moving from this side of the neuron only goes to the end and doesn't leak out and affect other neurons. Okay, so I'm getting a little bit ahead of myself talking about signals, but it's very important. So what the dendrites do is they take in signals. So let's say I have a neuron here, and so here's the neuron I'm looking at, and this neuron comes in and it says, hey, do the thing. So this is a positive neuron, and it sends a signal to this neuron saying, hey, do the thing. And it wants to get this neuron to start signaling. Well, what if you also have another neuron that's also synapsing on it? Cat, come on. My cat's trying to synapse. It's also synapsing on this neuron, but this is some sort of negative neuron. So this neuron is saying, no, don't do the thing. And you have this kind of conflicting signal where one thing says yes and the other one says no. How does the neuron know if it should then fire or not to get a signal across? Well, that's a good question, and that is what happens in this part of the neuron. This is called the axon hillock, or the hill of the axon, I guess. Sure. So the axon hillock, so by the way, the signal always moves in this direction, is where everything gets summed up. So in the axon hillock, all the signals that are coming in through the dendrites from other neurons are saying, should we do something? Should we not do something? And it has to hit the correct threshold in the hillock before the signal says, yeah, we should do the thing, and it zaps down the axon and then releases a neurotransmitter in the end. So what we're going to do is, now that we have kind of a basic idea of the anatomy of the neuron and kind of where it came from that general cell idea, we're going to look at what is happening exactly within the axon, because it's all about, so if this signal is strong enough to send the rest of the signal down the axon, that's called an action potential, okay? And what it does is it jumps from the start of one Schwann cell to the next Schwann cell, so these are cells, pew, pew, and this is called saltatory conduction. Okay, so saltatory, I don't think I'm spelling this right. I always remember it because it involves salts. It's in the slide somewhere. Conduction, saltatory conduction, and each one of these little spaces, so you'd think this would be a lot better conduction or a lot better insulation if, instead of having little like individual cells, this whole thing were completely encased in one giant cell. Well, it turns out these little in-between spots are really important, and they're called nodes of Ranvier, because that's the guy who discovered those. And those are where the signal gets kind of picked up and then sent along its way. And it's kind of like playing telephone, but like super fast, okay? So like the signal gets summed up in the hillock. If it's great enough to make the action potential happen, so that for the signal propagation to happen, it zaps along, pew, 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 to each node of Ranvier, and it gets kind of carried along each time. Otherwise, if that didn't happen in our longest neurons that go from our hip down to our toes, the signal might kind of run out halfway through, and then what do? You do nothing. Your signal's totally lost just halfway through the neuron. So in this case, it keeps it zapping along to make sure it gets to the end result. And we're going to talk about what's happening here at the synapse and what that really means. Yeah, let's go to some different figures.
Okay, so we've looked at this anatomy, and now you should be able to label the definite parts of the neuron. We've got the cell body, the dendrites, the axon. These are the Schwann cells. The intermediary parts are the little gaps are the nodes of Ranvier, and then this is the axon terminus. So to look a little bit closer at those nodes of Ranvier, if you were to take a cross section, you'll notice that each, sorry, not the nodes of Ranvier, but the Schwann cells, you'll notice that each one is one cell that wraps it, so there's a nucleus of this Schwann cell, and it wraps itself around the axon, and it just keeps wrapping and wrapping and wrapping. And the reason is why is the more layers of membrane you have, the more like basically gatekeepers that a signal or a molecule would have to get through in order to pass through this cell in order to leave the axon or to get into the axon and then cause a confusion of signal. I do want to mention that not all neurons look the same, so there's a high level of diversity in neuron shape and size and just interconnectiveness. We do kind of stick with this idea of the multipolar neuron because it's the easiest one to see the directionality of the signal. Like it's hard, excuse me, in a unipolar neuron to know which way does it go. I mean, if everything signals in the axon hillock, which is here, well, what's happened? There's no dendrite here, and okay, so don't even worry about that by just sticking to our one kind of generalized neuron that's got the dendrites here, the axon hillock here, the axon here, which you can see we have left off the Schwann cells here for simplicity, and then the axon terminal here. So I keep saying that the signal is important, that nerves are directional, they always move from the cell body, the signal moves from the cell body to the axon terminus, but what am I talking about when I talk about that signal? And so let's go over here and get a little bit of animation going on to make this a little bit easier. So if this is the neuron, the signal that actually travels is electric. It's an electric current, and it comes in through the dendrite, it gets summed up at the axon hillock, and then it passes down the axon. And this happens incredibly quickly. This happens in microseconds compared to what we can visualize, what we can see. This happens before we can even think about it happening because it all involves thinking, basically. Let's pause on this real quick. So when that happens, the idea is what is electric charge? So electricity is just the movement of charge. So if you've ever been shocked, like one time I was reaching behind a desk to get to a plug, and instead of touching the plug, the nice happy insulated part of the plug was sticking out a little bit, and I touched the tine and it was in the socket, and it shocked me, and I just immediately all of my muscles in my arm jerked because they were stimulated by electricity, which is what neurons do. Okay, so what they do is they take in some sort of a signal here. It's not electric at this point. They take in some signal here. That signal does something here, and then it shoots electricity down that axon, which then gets translated to a chemical signal here. So we have a chemical signal coming in at the dendrite. We have an electric signal traveling down the axon, which is incredibly fast. And then we have a chemical signal being released from the neuron. So how does this happen? How do we get this change at all? What is even happening? And this is the action potential, which you really need to know about. Okay, so let's keep this going. So let's take a closer look at what's happening inside the nerve. So if we look inside the nerve neuron, okay, cells tend to be a lot more negative than they are positive. Let's go back a little bit, okay? So there's a whole lot of positive charge hanging out outside the cell, and there's a whole lot of negative charge hanging out inside the cell because the cell is full of proteins, and proteins tend to be a bit negatively charged. Okay, so to balance that negative charge, you also have a whole lot of sodium inside the cell that exists as an ion, and it's trying to balance that. And in order to make sure that you have the right balance of sodium and potassium, you have these sodium-potassium pumps. And what they do is they trade 2 potassiums for every 3 sodiums. And so if we're doing that, we're establishing a sort of charged system here. Okay, so we're keeping that negative on the inside and the positive on the outside. But because it's more negative inside, those positives stick around outside, but they just can't pass through unless they have that sodium-potassium pump happening. So this sort of natural charge difference that gets set up between the inside of the cell and the outside of the cell is called the resting membrane potential, and that's going to become really important in a few minutes. So let's take a look. Okay, we're not going to look at that right now. Let's go back over here. I should just move this. All right. So we're going to go back and forth a little bit, but let's keep going. So the channels, so the neuron itself has, if this is the cell membrane of the neuron that we just kind of looked at, there's all these different kinds of proteins that act as pores or channels or voltage gated things, gateways, that will allow stuff to either come in or out of the cell depending on what the cell needs, and some other things we're going to talk about. The one you see talked about the most and that we're going to see a lot is the one that exists in the nose of Ranvier, which is called the sodium-potassium pump. So again, this kind of just puts out what I said. So because the cell is full of lots of proteins, cells have a negative charge, including the neuron. If we want to balance this, we have to put sodium chloride, we have to move sodium out of the cell. And that, in order to do that, there's this protein setup that will say, that says, okay, I will let you move 3 sodiums out if you give me 2 potassiums. This is going to make sure that the cell doesn't end up shrinking overall, because water is going to move where there's more stuff, and so water wants to leave. But it still sets up this really big gradient now where there's a whole lot more positive stuff outside than there is inside. And so that if the cell was just allowed to open up all its doors, so to say, all that positive stuff would just come rushing inside. And so what the neuron does, what it's evolved to do, is take advantage of the fact that the positive charges that are existing outside of the cell really want to come inside the cell due to diffusion. So every single cell in our body has a sodium-potassium pump. It's really important to maintain osmotic pressure and charge pressure. In the excitable cells, like neurons, muscles, and different glands, this becomes the cells have basically co-opted this fact to use it to its own advantage. And so there's something called, so if you've ever done any kind of electric work or electrician work, there's something called a voltmeter where you can measure how electric something is. And so if we measure the inside of a neuron compared to the outside, it has a resting potential of about -70 millivolts, okay? That's an important number. Make sure you know that, okay? So what's happening here, so when it's just chilling and doing its thing and nothing big is happening, sodium really wants to go in, but it can't. It's stuck outside. Potassium really wants to go out, but it can't. It's those pumps don't allow it. And so you have this charge gradient where the positive as a whole want to come inside. The potassiums want to be balanced both inside and out. So you have an even number of potassiums on the inside and the out. And so does the sodium. But right now all the potassium's inside and all the sodium's outside. And there's still way more negatives inside than there are outside. So we have this resting -70 millivolt potential inside the cell. So the inside of this neuron has a charge of -70. Awesome. So once a cell gets activated, that sodium just starts flowing in, boom, 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 boom. And if it's flowing in, it causes the, so if we started at a -70 charge and suddenly we start filling that cell with positive charges, then we're going to get an increase in that charge. And that's important, because the more increase in charge that we have, and when we have an increase in the internal charge of a cell, that's called depolarization. If a cell gets activated, why is this not spelled right? It doesn't seem to think, okay, whatever. If a cell is activated, then a whole bunch of sodium starts to flow in, and that's going to be really important. But in order to reestablish a balance, potassium is going to have to flow back out in order to correct for this charge gradient, and that's called repolarization. It's going to be important in a second. We're going to put together all of these things. So what happens is, if you have, let's say you have a whole bunch of signals. Let's see if this animation has it. Okay. Da, da, da. So there's sodium being pushed. Okay. Let's see. Da, da, da. It does not. Okay. So we talked a little bit about how you could have more than one neuron synapsing on this neuron. So you could have a whole bunch of excitatory neurons saying, do the thing, make that muscle contract, that are telling this neuron, do it, make it contract. If enough signal comes through and gets summed up in that axon hillock, all of a sudden you start to see sodium get rush into the cell, and it's going to do it at each Schwann cell. Okay, so you have this kind of jumping of charge where sodium starts to come in, and then it starts flowing down that neuron in that kind of zappy way we think of when we think of neurons. Okay, so if so let's imagine that enough signal has summed up here that all of a sudden that signal can go zapping down the axon. Awesome. That only happens if enough excitatory signals have come in to say, yes, we want this neuron to go. On the other hand, there are also inhibitory neurons that could also synapse on this neuron to say, no, don't do anything yet. And so they're releasing their inhibitory signals. Then you have other ones that are releasing excitatory signals. It's only when you get to a certain threshold that this action potential will actually happen. And so this nerve is not going to be able to release a signal, that zap that goes down the neuron, unless that threshold is met. Okay, let me go back.
over here. Okay, so when that threshold is met, you get the action potential, which is a very fancy couple of words that just means the signal moves down the axon in a very specific way. You have the sequential change in the permeability of the cell. What does that mean? That just means those gatekeeper portal pores start opening up. So if you're thinking about those Schwann cells, so if we go back up here, so if this is the axon hillock and it says, yes, let's fire that signal. So at each node of Ranvier, you have sodium channels open up. So remember, sodium's existing outside here and it really wants to move into the neuron. So every time each one of these little places, you have the sodium channels opening. So, oh no, all these sodium channels opening, and then the signal zaps in, and the only way it can go, because it can't go this way, there's too many proteins, is this way down the neuron. And so you have the sodium signal go, and it zaps to the next node of Ranvier, where more sodium channels open and allow more sodium to come in, and it zaps to the next one, and the next one, and the next one. And so that's what saltatory conduction is. That's where you have this flow. So if this was one axon over time, you would see sodium traveling in this little pathway. But behind sodium, so it's depolarizing the cell by making it become a bit more positive as it zaps from node of Ranvier to node of Ranvier. Behind it, though, the cell's like, oh no, what did we do? And it starts pumping out that sodium and pumping in potassium in order to try and balance out that those charges, okay? And so basically you have repolarization coming along behind the sodium to make sure that, hey, we only want the signal to happen once if it's going to happen. You don't want to have multiple zaps going at the same time because you might get an overstimulation of the signal and you don't want that, okay? So in the neuron chain of reaction, blah, blah, blah. Okay, so, and I think I said, so in this case, so sodium's moving in, which is making it really positive. So in order to balance that, potassium gets moved out because you're trying to balance that positivity without making sodium move backwards through the neurons. Because if this started moving sodium out here, well, this sodium would try and move in both directions instead of just in this direction. So that's important. Okay, so why does that actually matter? Okay, so I talked a little bit about excitatory and inhibitory neurons. So action potentials have to be initiated by a stimulus. They must be appropriate and they must be big enough. If someone just sort of lightly touches you, you're just gonna, depending on the situation and what's happening, you may not react at all. You may just be like, oh, what is that? Is that a bug? I don't know. But if someone smacks you really hard, that's a really big stimulus, and that's enough of a stimulus to cause something to happen. If nothing else, if it's hitting just right, you could have a reflex arc. If it's hitting somewhere else, you turn around and see what's happening. Whatever it is, anytime you have a big enough signal happening at the right time, you end up with an activation of the neuron. Now, a lot of times neurons can be primed. Like if you go into a haunted house and you're going to be scared and you're really excited about being scared by the haunted house, then stuff that wouldn't normally be scary will scare you because you've primed your neurons to be appropriately stimulated by certain responses. Or if there's lots of different examples that we won't go into. So in order for an action potential or a nerve signal to be transmitted, you have to have the right signal and it has to be big enough. So in neuron, we use the example of reflexes a lot because neurons can be activated by physical stimuli like pressure whenever you're hitting something. We think about heat also, or the lack thereof, being a good stimulator. If you're too cold, if you're too hot, if you're just a little cold or a little hot, anything like that. Light is a good stimulator. There's a lot of studies. Someone just won the Nobel Prize for how circadian rhythms, or the amount of sunlight you get per day, is so important in regulating everything else in your body. So light is clearly a very important stimulus, motion, etc. Just there's all these physical things that can trigger a neuron. There are also chemical triggers like acetylcholine. So acetylcholine, remember, is the neurotransmitter that gets released by the neuron onto a muscle. And so there are other ones like dopamine and oxytocin, all these other neurotransmitters, serotonin, that we know are affected by depression and anxiety. Then a lot of these neurotransmitters we only know about in disease states or in
pathological states where something bad has happened, and suddenly people are like, oh, we should pay more attention to this because this is not a good state to be in. That's how we tend to discover these kinds of things. But so that's why if you are giving somebody a certain, or if the way certain drugs work is they go and they act as neurotransmitters and cause a stimulation of a neuron or block certain neurons. So let's say you have that stimulus. So what is happening? And this looks, you might be tempted at first to say this looks a lot like a heartbeat, but it's not quite. This is what's happening in a neuron if you were to look at the voltage, okay? And this is very important, hint, hint, what is happening in an action potential. So if a stimulus is big enough, that neuron is going to hit that threshold I was talking about, that threshold voltage that that axon hillock has to get to in order to start propagating a signal down the axon is negative 55, okay? So the cell just kind of chills at negative 70. You could have one or two failed signals that are saying like, hey, neuron, do the thing, but you have to get a big enough, most appropriate signal that depolarizes, brings in enough sodium to get the threshold to negative 55 before you actually see that huge shooting of the signal down the pathway, okay? So, or down the axon. So this is the threshold here, or sorry, this is the resting potential. So you'll see resting state or resting potential on both sides. So that's the nerve starts at rest, it ends at rest, okay? You have to hit the threshold before you suddenly see that huge influx of sodium. That's the depolarization stage. Once that gets to a certain point, the action potential is triggered and it starts shooting down the neuron, okay? So while that's happening, if we're just kind of watching one place, you see a repolarization happen. And so at this point, you have, this is your sodium channels opening up. Because you get to this positive 40, okay, so we go from negative 70 to negative 55 to positive 40, that's such a huge change. That triggers what are called voltage gated channels. And those are the ones that open up and make potassium start leaving the cell, and that causes repolarization. That repolarization is trying to bring the neuron back to its original resting state. It actually overshoots, and you get this refractory period, which makes the neuron non-responsive for just a little bit of time, which is good because you don't want a false signal to make your neuron shoot off when it shouldn't, okay? And so this is caused by the neurotransmitters usually causing the depolarization, that reaching that threshold. Then that threshold, once the threshold is met, you have this huge depolarization caused by the opening of sodium channels. Once you get to negative or positive 40, you get the voltage gated potassium channels opening up. So they're voltage gated. That means they open at a certain voltage, and that certain voltage is 40. And they let potassium rush in the cell. When they do that, they let potassium leave the cell. So they let so much potassium leave the cell that the cell actually gets way more negative than it should. So it gets down to like maybe, who knows, negative 90 or something. That's not an important number to know. Just know that it goes, so different neurons are going to go to a different threshold based on if they're a fast twitch or a slow twitch kind of neuron, if we're talking muscle neurons. So that refractory period, though, causes them to reestablish their resting state before they can be stimulated again. All right. So here's a good point. If the stimulus is not big enough, then the neuron will return to its resting state. So this is if it's only slightly cold, then and your sensory neurons say, hey, it's sort of cold. Your brain will say, yeah, it's not really that cold, whatever, and not get up and make you put on a sweater or cause you to shiver or whatever. So that's kind of an easy way to remember it. But there's so many more examples. Okay, so that's all awesome. So that was that electrical signaling. What happens at the end of this electrical signaling? So once you get that increase in voltage, the axon action potential causes, once you get down to the very end of the neuron, so that would be, let's see, here. So we have the action, so remember this was a chemical signaling coming in, this is the electric signal, and it it concludes in another chemical signal down here. So what's happening? So way down there, where are we? Here we go. At that axon terminal,
you have these vesicles, and these vesicles are just hanging out. They're full of the neuron-specific flavor of neurotransmitter. This could be acetylcholine, if this is synapsing on a muscle and it wants the muscle to contract. It could be dopamine, if it's, for many different reasons. It could be oxytocin. There's some TED Talk of this guy who calls himself Dr. Love, and he says, all you need in life is 8 hugs a day because it releases oxytocin, and that's all you need to be happy. That's false. Remember how these signals have to be in the right, you have to be in the right frame of mind in order for that kind of thing. If a stranger walks up to you on the street and wants to give you a hug, you're not going to be as happy as that guy wants you to think, okay? So neurotransmitters are really complicated, and anyone who, there's a lot of memes out there that say, oh, oxytocin is happiness. Well, oxytocin also leads to stalker behavior. It's an attachment hormone, which is really important in establishing bonds between loved ones and mother and child and that kind of thing. But it's also, in bad dosages and bad timing, leads to stalker and murderer behavior. So, again, this is another example of things being incredibly complicated. Okay, but the point being of all of this is once that action potential reaches the bottom of the axon terminal, it causes those vesicles to go and bind to the cell membrane here, and it releases those neurotransmitters into the synapse. So the synapse, or the synaptic cleft, is this open space, and it seems like a bad design at first, except that it means that these have to travel in one direction in order to cause a stimulation, and we'll not get into it, but these are now neurotransmitters. So we've taken an electrical signal and we've ended up in a chemical signal, and those chemical signals go and they bind to the dendrites on the next neuron, and they cause an excitatory or an inhibitory response, which then gets summed up by the next axon hillock, etc., etc. Things go on. Okay, there are a lot of antidepressants, and I talk about this a lot because it's just a good example. There are a lot of antidepressants that what they do is there are these things here, these purple things that I guess that's what these are representing, which are reuptake proteins. And what they do is, the body tries really hard not to waste anything. So if you have a bunch of neurotransmitters hanging out in the synaptic cleft and they're not being absorbed by the postsynaptic neuron, well, this neuron wants to take them back up. Like, oh, if you're not going to use them, I'll save them for the next action potential. But in the case of depression, there's a lot more of these uptake neurons or proteins than there are the postsynaptic ones. And we need, when you're in a depressed state, you need that neurotransmitter to get to where it's supposed to go as much as possible. So a lot of antidepressants are reuptake inhibitors. So what they do is they go and they block these channels, which allows a neurotransmitter to stay in the synaptic cleft longer and be more likely to go and stimulate the next neuron. So you hear about serotonin reuptake inhibitors, dopamine reuptake inhibitors, etc. Okay, so here's kind of another, this is a very good thing to know about. Here's another a way to look at the action potential in steps. So if you're looking at a neuron when it's in its resting state, it's resting at -70. It has some set of stimuli that are going to make it, if you have enough stimulus, it's going to cause it to reach that threshold of -55. Once it hits -55, so that's the threshold of excitation, or just threshold. Okay, so we have resting state, and then we have the threshold. Once you hit the threshold, you open up a whole bunch of potassium channels to make potassium leave, and a bunch of sodium channels that just start flooding like crazy. Okay, so that sodium starts rushing into the axon, causing the signal to go down the neuron, zap, zap, zap. Once you get to, this says negative or +30, somewhere between +30 and +40, you get that peak action potential, and it causes voltage-gated channels to open, and it causes repolarization to start happening. So this is depolarization, this is repolarization. So now you're moving all that positive charge out of the cell back into the extracellular matrix, but you do it so much that you actually get that hyperpolarization, or that refractory period, where it's overly negative, okay? And then you have to let it go back to normal and back to resting state. So hopefully that's another way to kind of understand that. Let's talk just a little bit about, so that kind of jumps a little bit, but let's talk a little bit about some other neurotransmitters. So a neurotransmitter is just a chemical stimulus for neurons. It's kind of, there are a lot of hormones that also act as neurotransmitters. There's a lot of drugs that act as neurotransmitters. They bind to those receptor proteins on the dendrite of the next neuron. There's a huge diversity of them. They can be amino acids, they could be small proteins, they could be lipids. There are gases. So in the case of erectile dysfunction, what's happening is the nitric oxide that is required for an erection is not getting to where it needs to go. And so stuff like Viagra is allowing that neurotransmitter to get to where it needs to go to cause erection. Sometimes even ATP, that energy currency molecule, can be a neurotransmitter. This signal, if you want to stop this signal, so the action potential is stopped, but you don't want any more, you've already done the thing, you want it to be over, you can reabsorb that neurotransmitter like we talked about, or there could be these other enzymes that get released by the postsynaptic neuron, which hydrolyze or destroy those neurotransmitters. So in a muscle, if you're trying to stop a muscle contraction, if it's a muscle that doesn't need to contract for very long, you have the acetylcholine esterase. Holy cow, acetylcholine esterase. So that's an enzyme that gets released by the muscle, and it gets released into that neuromuscular junction to break up all the acetylcholine it doesn't need anymore. So here are some good examples of neurotransmitters to know. Each one of these have so much more information on them, but we just don't have time to go into them, but they're so fascinating. Acetylcholine is one you should definitely know because we talked about that with the the neuromuscular junction. That's the one that causes a muscle contraction. Norepinephrine is, it's a mood-altering it causes you to go into a resting state rather than a stress state. So it's it's not on here, but we're going to talk later about epinephrine or adrenaline and how it causes the fight or flight state. Norepinephrine helps you go back into the rest or digest state, the safer state. Dopamine also can alter mood. This is also affected by a lot of drugs. Serotonin is what helps us sleep. And so if you don't, this is usually when people say that, oh, I had a lot of turkey and now I'm really sleepy because of the tryptophan. No, you're sleepy because you ate a lot of food and then you started releasing serotonin, which makes you want to go to sleep so you can digest it. And so it's not the turkey itself that makes you sleepy, sorry. GABA is an inhibitory neurotransmitter. If you have a lot of GABA neurons synapsing on something, they're trying to stop something from working. And then we have these endorphins and enkephalins, which are endogenous opioids, so that's a lot of E words. So what that is so endorphins are what are released when you experience a whole lot of pain. So like if you're getting a tattoo and your body is basically saying, oh my God, this is a painful situation, get out. And you're not getting out, so your body says, well, you're not responding to my stress response, so we gotta just deal with this. I'm gonna release these endorphins so you feel less pain so that you can get out of this situation. So that's what endorphins and enkephalins do. They are natural heroin for your body. They're endogenous opioids that will let your, how do you even spell that? They're endogenous opioids that will let your body deal with a stressful situation. A lot of people who are stimulus seekers, who like to, base jumpers or paragliders or that kind of thing, they are not, they could be, but maybe not as seriously be addicted to that feeling of that endorphin rush. So that's what that stimulus seeking is all about. There are, of course, a couple of really bad diseases of the neurotransmitter systems. So like tetanus, so we have to get tetanus shots every 10 years, especially if you hit a rusty nail or something. What it does is, so tetanus is a toxin released from bacteria, and it inhibits the release of inhibitory GABA. So if GABA goes and causes neurons to stop being, or muscles to stop being contracted, if you don't have that signal that says stop contraction, then you're going to stay contracted. You're going to go into rigor mortis without actually being dead, and that's what's happening in this very classic painting of one of the first examples of tetanus. All of your muscles, the first thing that happens is the muscles in your jaw get affected, and so you can't stop clenching your jaw, and that's called lockjaw, and it's one of the first signals that you have tetanus. It can be treated by GABA, but you just get you get on antibacterials to get rid of the bacteria, and then you have to take GABA in order to undo all of this. There is another toxin called botulism. This is what's in Botox, and it's released from bacteria that actually inhibit the release of acetylcholine. And so what this does is it means you can't contract your muscles. And so while this will let you stop contracting, botulism causes you to be unable to contract. So this causes flaccid paralysis. So this is an example of a kid who has it, and it's very sad, but people inject it into their faces all the time. And in very, very small doses, it causes your muscles not to contract in your face, so you don't get wrinkles as much. So that's part of, yeah, we'll leave it there. All right, so if this were a test question, what is happening here? What's happening at A, B, or C? So this is our resting potential. Here are some failed attempts to have a stimulus happen. Oh, and then depolarization happens. So B is depolarization. And then we get to that high threshold of the action potential, and then the voltage gated channels open, and then repolarization happens. All right. So, and I just answered both of these. Okay, this entire event is called an action potential. This is happening in the axon of the neuron. It's started by those neurochemicals being released by the synapse and going to the dendrites of the postsynaptic neuron. And the end result is the release of neurotransmitters in the next, into the next synapse. Okay, so it's kind of a series of a release of neurotransmitters. Okay, so here's the word that I couldn't spell earlier, and even my, of course, Word doesn't like, or PowerPoint doesn't like spelling it, but saltatory conduction is what I was talking about when you have a signal jumping from node to node. And so I like to assume it's because it involves salts like sodium and potassium, but that's probably not it. But all it means is you have this conduction of, you have this jumping of signal. So this, between the nodes, the signal travels really fast, but then it starts to slow down as it starts to approach the next node, but then it's okay because it gets recharged and pew, pew, pew. It just zaps down, and that's called saltatory conduction. This can allow those neurons to travel at 250 miles per hour, which is incredibly fast and very, very important. Okay, so we talked a little bit about myelin. So the Schwann cells, what are they doing? Myelin, so sometimes those Schwann cells are called the myelin sheath because they create this, if the neuron is a sword, then the myelin is the sheath that surrounds it. They wrap around the axon, and they have just so much lipid layers, so many lipid layers that will so remember that it's really hard for charged particles to get through lipids because it's hydrophobic. And so if you have just, it's hard to get through one layer of lipid if you're a charged particle. So if you have a ton of layers of lipids, it's going to be basically impossible for a charged particle to leave. And so that means the signal stays pure going down that axon. So again, in something like Parkinson's disease, these Schwann cells break down, or they're very, very, very thin, which allows signal to leak out of the axon and then interfere with other axons around you. So you get signals overlapping in neurons. And that's why you see the kind of characteristic jerking in Parkinson's disease. We're going to talk next lecture about the oligodendrocytes, but they're basically in the central nervous system versus the peripheral nervous system, which we'll talk about in the next module. You have different cells performing the same function, but it's myelin. Myelin is the lipid that is actually causing this sheathing behavior. It is a lipid inside of those membranes. Another thing to remember is that white matter has to do with those. So when you say white matter versus gray matter, so we call our brains gray matter. They're mostly that unmyelinated axon, or really the neurons, those cell bodies. Okay, they're not covered. So if you look back at that neuron picture, it's not covered in. Let's go back up here. So this part doesn't have myelin, and myelin tends to be kind of white. So this we call gray matter, even though technically it's kind of pink. I think they called it gray matter when anatomists were first looking at cadavers that have been dead a long time, and then it would be gray. It's pink in a more natural, healthy brain. But so the brain is full of cell bodies, so many cell bodies, not a lot of axons. The axons leave the brain and go traveling down the spinal cord to the rest of the body so they can send that signal. So the spine and the rest of the body are made of white matter, while the brain is going to be gray matter because it's mostly the cell bodies. All right, that's all for this lecture, and I'll see you in the next one. Bye.